Very good evening, good afternoon, good morning from wherever you are listening to this program today. I welcome with my heartiest cordiality to the International Conference on Dental Genetics and Clinical Diagnostics, Chapter 2. So, today we are now embarking on this journey for the 3 days journey and a small bit about the conference. This conference is an endeavor to spread awareness about genetics, research, genomics, and different other new arenas of science and education. So, in this conference this year, we have three focus areas. One is genetics and genomics, second focus area is artificial intelligence, and third focus area is research career. Well, as you see this session embarking, we have an industry specialist and a very famous personality and from a very famous company known as Premas Life Sciences. Sorry. So I have Debjani Shaha, our first speaker, and I'm very grateful to Debjani for making time in spite of her busy schedule. And now we will embark on a fantastic journey through her session where she will enlighten us about the different arenas of omics, that is whatever you can think of when it comes to omics, DNA, RNA, everything, and into the realm of saliva and the future applications and what is happening in this entire world with this fantastic area. So without any ado, I would like to call in my guest and our first speaker of the day.
Thank you so much, Gargi, for inviting me to be the opening speaker of this workshop series that you've embarked on. And I'm so excited to be presenting on a technology that has been really close to, our hearts. And, we've really kind of toiled hard to increase the niche for next generation sequencing technology in the space of both the research and clinical domains in the Indian market. And since this is the opening talk, I just thought we'll start with a brief overview of next generation sequencing for the benefit of all the attendees, and then we can go into the specifics of the omics, right? So,
Definitely.
without much ado, let me introduce myself. I'm Devjani, and I oversee marketing and product management functions in this company called Premise. Now, just a couple of lines about our organization. So, Premise is primarily a life sciences devoted a life sciences devoted organization, and we incepted as a contract research organization several years back, about a decade and a half back. And we also spun off to what we call Primas Life Sciences, which is basically, a firm which brings these cutting edge technologies in the life sciences space to a lot of laboratories. And formerly, we were only operating the space of research and development. But, as we go ahead in our times, we distinctly see that there is a paradigm shift of
Right.
next generation sequencing based applications in the clinical domains, right? And and I'm here to talk a little bit about the technology and what are the implications, what are the applications in the clinical sphere. All right, something very basic. What is next generation sequencing? So, as many of us, would have actually done sequencing in our lives and sequencing in our laboratories. And if we just basically look at, how sequencing has evolved, uh, a lot of us used to do Sanger, in the very initial days of our research, and then slowly the market has shifted to next generation sequencing. Now, there are distinct advantages to it. And essentially, what are the advantages? Uh, NGS provides digital data. That means each read is analyzed independently, it's quantitative. And what you're trying to do is through overlapping reads, we're generating very, very quantitative and high confidence variant calling. So any variant that's there through the advantage of multiple reads is nicely flagged out. And when we actually talk about next generation sequencing, we're talking about massively parallel sequencing. That means to the range of anywhere between 100x to about 1000x, and sometimes maybe a little lesser. But the fact remains that you're actually talking about millions of Sanger reactions happening on a single chip, which basically lead to the detection of variants which are present at sometimes very less abundance, right? So when you actually talk about Sanger and NGS, one of the biggest differences between the two types of sequencing is the fact that the limit of detection in Sanger is at about 20%. That means anything less than that is not nicely flagged out, right? However, in a lot of pathologies, we see that the mutant allele is in a very low abundance. That means anywhere between 5% or maybe even less than that. So the limit of detection of NGS is much lesser than Sanger sequencing. Therefore, it makes it possible for the researcher or for the clinician to be able to decipher mutations which are present in lesser amount of abundance. And that is the biggest advantage of NGS, apart from the fact that you can scale up your sample numbers. And that's really essential in today's times because you having a lot of samples for a disease analysis. So essentially, you can upscale the number of samples that you can actually study. Besides of the fact that you can also throw in a analysis for a lot of genes together. So you can analyze a lot of genes for a lot of samples together with the advantage offered by NGS, right? Uh, Also, when you are actually talking about disease, there are a lot of genetic drivers of disease. And NGS is a very, very sensitive method to be able to quantify or qualify the genetic drivers of disease. And you do it through multiple modes. You can study copy number variants, you can study gene expression, regulation, protein DNA interactions, epigenetics, metagenomics, and now with some of the recent technologies, you can even study proteomics using NGS as a analyzing technology. So there are multi-pronged advantages or multi-pronged ways in which NGS can be used. It's basically a means to an end, the end being the evaluation of a specific molecular basis for a disease, a pathology, or a biological effect. And this is basically some of the advantages of NGS. It's hypothesis free, it's highly sensitive, it's got a very high resolution. You can multiplex a lot of samples, and it supports a bunch of biological interrogations as what happens in the body, right? Uh, these are some of the differences between the popular methods like qPCR or Sanger, which have been hitherto used, and the paradigm shift, as I said, is towards next generation sequencing for a very good reason, that it's got a enhanced discovery power. It's got a better resolution, and like I said, you can actually study information from multiple nucleic acids. Now, typically, what is the workflow, uh, like involved in the next generation sequencing, in a particular next generation sequencing experiment? First of all, the biological sample has to be processed to make sure that the nucleic acid is available from the biological sample. It could be blood plasma, it could be saliva, it could be environmental sample, it could be stool, it could be tissue biopsy, anything. So you have to get the nucleic acid out. You have to process the nucleic acid to get it into a format which is compatible with the sequencer, because you can't load the nucleic acid as it's. And then finally, you have downstream analysis to basically derive important interpretation from the sequencing data that you get, which I believe is the most important part of the entire experiment. And typically, when you actually say you process the nucleic acid, that means that you chop it up into pieces using either a mechanical or a enzymatic fragmentation method. And then you append known sequences at both ends of the DNA fragment, right? So you have, these two ends which are appended with some known sequences which are known as adapters. Now, What are these adapters? What is the function of the adapter? And the adapter hosts a region which is called the sequencing primer binding region, another region which is called the index, and the P5 and P7 adapters. Okay. Now, what is the independent function of all of these little pieces? The sequencing primer binding site, as the name suggests, is the site where the primer for studying the sequence it comes in bytes. The index helps us to be able to throw multiple samples in a single lane of the sequencing chip, and the adapter helps us in something in a process which is called cluster generation or clonal amplification of the template. That means this entire template is made multiple copies of, and this entire process happens in an automated fashion on a sequencing chip. Okay, so the reason why we need this clonal amplification to take place is to be able to amplify the signal which is being generated with a single molecule, and you amplify it multiple times so that the detection systems which are required need not be the size of a house or even bigger. You can actually be able to detect those signals from a small benchtop, right in your laboratory, right? And like I said before, one of the biggest advantages of next generation sequencing is the fact that you can actually pool multiple samples together. And what makes it possible? The moiety in the adapter that makes it possible is called the index. Now, what happens is when you actually have multiple samples, in this case, you have 10 samples, all the fragments coming out of a single sample will carry the same indexes. So that when you actually demultiplex the reads, those reads can be binned by the indexes into the multiple sample types. And so you can classify which fragment comes from which sample. All right. Now, the company that probably has the biggest share, approximately about 80 to 85% of the global share of sequencing is a technology called Illumina, and they operate by a sequencing method which is called sequencing by synthesis method. Okay. Now, what is sequencing by synthesis? It means that the new strand is being synthesized, and as it's being synthesized, the sequencing happens. So, assuming that this is the template on which you have the adapters appended on both the sides, when I said before that these adapters help the template append to the sequencing chip on which you have a lawn primer lawn, and you have these primers which are complementary to the adapter. And through a series of amplifications which are known as bridge amplifications, you have these clonal clusters of each of the fragments created. And this clonal cluster, I mean, the biggest advantage of this is that this is able to amplify the signal generated through the addition of the nucleotides. So when the nucleotide the incoming nucleotide is added to a growing strand, then the same process happens over all the fragments in the clonal cluster. And this basically amplifies the signal, and you're able to decipher, what is the nucleotide, which is basically complementary to the incoming nucleotide, right? And when you actually say SBS, it also means that all four different types of nucleotides, that is the ATGC, are present in the same medium, right? And so basically, there is a competition between the nucleotides for getting added to the, extending strand. And this competition ensures that whatever best fits to the complementary base on the parent strand is the one which is incorporated, and you don't have a bias created for any specific kind of a nucleotide, right? And since these nucleotides are all carrying these different fluorophores, you're able to essentially gauge what fluorophore is getting added and therefore what nucleotide is getting added. And so these, fluorescent signals are generated, which is then converted into the base calls. Right. So this is the simple process for sequencing. If you want, there are a lot of nice and educative videos present on YouTube and on company sites which explain this process further. Now, once your sequencing is done, you have like a map of the sequences to the reference gene, right? And depending on what maps, you have the variant or genotype calling, annotation and filtering, and the functional effect called out. And eventually, you have like the analysis or interpretation of the sequencing data. And, at the moment, there are plenty of tools which are available. So essentially, in the current times, the sequencer, I mean, there was a time when, like when you actually had to sequence, you had to devote a room full of instruments, and each instrument would be responsible for different functions associated with the sequencing process. However, in the current times, you have these benchtop sequencers available, which can pretty much do everything by themselves. So, like you have the sample prep, which is, like upstream to the sequencing process, which is off the instrument. But starting from the clonal amplification to the sequencing process to the, like the secondary data analysis, everything happens on the sequencer, right? And beyond that, sometimes like the tertiary data analysis happens on a lot of cloud platforms which are available. And sometimes you can actually have these third party pipelines available, some of which are paid, some of which are, like which come with a certain cost. And once you do that, you can have the annotations done and reports generated. And hence, for every species, you can actually study a bunch of applications, right? Especially for human genomics, I think the list of seq applications is growing all the time, but you also have a bunch of seq applications for other species, right? So, and that includes studying the DNA or RNA or, like, so you have the whole genome, exome, transcriptome, metagenome, epigenome, etc., etc. And even like I said, in current times, even the proteome can be analyzed by a downstream analysis method, which is genomics related, right? So there's a bunch of seq applications, and based on what you want to do and what your application is and what your end objective is, you can pick and choose. Right? Now, like I said before, earlier, like when somebody had to do a sequencing experiment, you needed to have like a bunch of platforms and you needed to have a really big space allocated to it, you know. But at the moment, in current times and over the last decade, what has happened is sequencing has evolved so much that people are actually wanting to turn the entire exercise into a very user friendly workflow, which involves basically compressing all the functions into what they call as benchtop sequences. We also have retail scale sequencers, but that is also like that's also increasingly evolving to be like a one box affair, right? So essentially, what does the benchtop system bring to your table in terms of the value proposition? It's really fast turnaround time. So essentially all you have to do is prepare the sample and you have to throw it into the sequencer, and the sequencer will do everything else, starting from amplification, clonal amplification, to sequencing, to the monitoring the run. So basically it's like load and go. It's push button and walk away. And it'll also give you a better flexibility and control on your experiment. And even if you don't have too many people devoted to running the experiment, it's very user friendly. So essentially, you can make do with a few people in your lab who can take care of the various aspects of the sequencing process. And ultimately, a lot of technologies, including Illumina, are increasingly laying a lot of stress on this end-to-end value proposition. What does that mean? It means that right from the sample prep solution to the sequencing solution to the analysis solution, everything is integrated with one provider, so you don't have to run to 10 stops to basically put an experiment together. Now, this was the background about sequencing per se. I'll spend the next 15 or 20 odd minutes to explain how sequencing is basically increasingly showing a paradigm shift in its applications in the clinical segment. And to start this segment, to start this portion of the talk, I just thought I'll briefly show snapshot of the clinical market for NGS in 2021 versus 2022. And now, if you just see the clinical market in terms of the various applications in which NGS is currently being used, some of the clinical applications are, and these comprise the major applications for which NGS is being utilized. Uh, and the biggest segment in that is HNA and EXO, right? And followed by NIPT, followed by small oncology panels, and followed by CGP. So, contrary to what we feel, oncology might be the biggest driver, it is the transplantation market and, to an extent, the genetic diseases and the reproductive genomics segment, which is probably the biggest driver of NGS in the last few years, right? And so, what are the major market drivers for NGS? It is A, acceptance of NGS in molecular diagnostics. And specifically after COVID, I think we've seen a huge deployment of next generation sequencing in the clinical segment. Precision oncology, it's increasingly being accepted, although there's miles to go before you actually reach a space where you can say there's optimal utilization. That also has certain challenges, but precision oncology is a major factor. Traditional testing methods, which are very tissue intensive. So, like a lot of pathologies, it's very difficult to get tissue. And so, traditional testing methods have an increasing tendency to shift to NGS because NGS is, that way is judicious with tissue. An accelerated adoption and utilization of NGS in clinical applications. Popularity looming around in non-invasive testing. And so the later part of my talk, which deals with salivary or oral genomics, is a classical example where non-invasive testing, liquid biopsies, and especially NIPT, have actually cemented the stage for non-invasive testing. Medical tourism, of course, and an awareness or sample availability, better logistics, etc. I'll start a little bit from oncology because, like I said, one of the major segments where it is showing a lot of promise is oncology, and it also has a good, kind of a, what do you say, connection with what we'll come to later in the talk. Now, as we all know, our genome is dynamic. Unlike what was the belief earlier, it's not static, right? And in cancer, especially, one snapshot is not enough. That means you're born with a certain composition of the DNA, and through our lifetimes, we accumulate certain changes in the DNA depending on what we're exposed to and our nutrition, etc., etc. And eventually, sometimes these somatic mutations reach a critical threshold or happen in certain genes, and that kind of leads to a deregulation of the way the genome functions in the body and leads to cancer. And eventually, it all spreads to reach a stage called cancer metastasis. And then, suppose if you are able to administer a therapy which causes a remission, sometimes the cancer is returns back because you still have a small a percentage of the mutant alleles thriving in the body, and then those bring a relapse of the cancer. Okay, so essentially there is an entire journey that the DNA undergoes, right? And so in molecular oncology we have a few goals. So what are these goals? A, first of all, risk assessment and stratification. screening and early detection. I think especially in cases where there's a familial history, you would want to be able to at least screen and make sure you flag off the cases which look ambiguous. Diagnosis, characterization, diagnostics, and monitoring. Right? Now, like for a very long time, iterative testing was done. So basically, you have pathology, then you have a tissue available. You take that tissue for certain molecular analysis, microscopy, like IHC, iterative gene testing, where you just end up testing one gene, that comes out negative, you test another gene that comes out negative, you test another gene. And oftentimes what happens is, you get a series of negative and bam, you run out of tissue for basically going to the test, which will give you some information, right? Now, the pitfalls of iterative testing. So, like um, the positive for iterative testing, if any, was the fact that you sometimes, if for the first time you get a positive, you sometimes escape off with a smaller cost. However, there are more pitfalls. What are these pitfalls? Typically, the testing time is long. There is a cumulative cost, and often you do not have enough sample to sometimes come to the hit, to the molecular hit, right? So suppose if you're able to combine the multiple genes that need to be tested into a single test and devise a multi-gene assay, and that has what NG has done really in the scheme of things. They have NGS through the availability of multi-gene NGS assays have basically reduced all of these things. It has reduced the turnaround time, it has reduced the cost, it has reduced the demand on the sample, right? So, in one sampling, you're able to get information on the multiple genes, and the chances of getting a hit through that is much more, right? So, again, some of the key drivers for the adoption of NGS in oncology is a better detection of common alteration, which is often missed by standard of care testing. You end up detecting rare genes like certain exons or certain fusions if you're studying the transcript, etc. You, like, suppose if you're going in for a comprehensive assay, you're able to find out some of the newer biomarkers like the tumor mutation burden, microsatellite instability. Like you're able to basically get a place into the clinical trials for some of the drugs. There's a lot of R&D happening in oncology, like at least 150 plus products are present, 150 plus drugs are present in the stage 2 and stage 3 clinical trials. So that is basically driving the use of next generation sequencing in multi-gene tests, in through multi-gene tests, physician behavior. So I think many of the clinicians are coming around to the awareness that it is a useful technique and has a good diagnostic potential and patient outcomes, better patient outcomes, wherein we've seen effective, management of the disease through, a good diagnostic, um, what do you say, a good diagnostic implementation. Some of the key barriers, however, would be data across cancer types. Report interpretation often is a challenge because the clinicians are not trained to do it, and a lot of times there is non-uniformity in report interpretation and a basic awareness and experience in next generation sequencing because it's such an esoteric test. Logistics issue, you need to have batching for some of the chips. So you can't just, like, if I have one sample today, I can't run one sample. I need to have a batch created, and that kind of sets the time back a little bit. Tissue availability, and often the quality does not really qualify for the NGS testing. Affordability, of course, because in India we don't have like a reimbursement through insurance companies. and the access to drugs in clinical trials. So even if you get information about a drug, it's not necessary that that drug is going to be available. Now, a lot of understanding that we have about the various tumor types or the various cancer types is through large scale global cancer research. So, like for instance, TCGA and ICGC have, like really contributed to our understanding of the various tumor types. And across the various countries of the world, they've worked on different kinds of cancers, and those are listed here in this slide. Uh, and through this, R&D, the information that's gone into the compendium has greatly basically contributed to our knowledge about the different cancer types. And India, very interestingly, has been a part of the oral cancer consortium, right? Institutes like NIBMG and ACTREC have done a lot of work on the Indian samples and have, you know, got such good publications. And through them, we know about the data for oral cancer, which is quite prevalent in the population. That brings me to the oral squamous cell carcinoma, the OSCC, where the early diagnosis has been difficult, and it's basically because of the lack of an effective OSCC biomarker, an efficient molecular validation method, right? Now, interestingly, saliva as a biological specimen, uh, has been tested for certain biomarkers. And if we're able to establish these through subsequent studies, we can actually use it as a non-invasive sample for, an effective screening and detection of OSCC, because phenotypically patients only come to the clinic at a later stage, right? And saliva sampling also provides a it's very easy to collect saliva, even for large scale sampling. So if suppose you want to study epidemiology, cancer epidemiology, which a few institutes in India are studying, and you want to do a longitudinal study, it's a sample which people will not hesitate to give, because it doesn't hurt to collect saliva sample, and you can do a large scale screening, and you can do a large scale study. And some of the biomarkers which have basically been flagged off through this, I would have loved to, like put a couple of studies from India also, but I have not done it for the interest of time. But I can share it offline, or we can probably catch up on them offline whenever, like suppose if somebody is interested. But as we speak, a lot of a few genomic, epigenomic mRNA markers have been flagged off for studying oral squamous cell carcinoma, right? And those are listed nicely, and there are some more also, which have been interrogated, through various studies. And as you can see, it's basically you know pointing at a multi-omic approach to study the disease or study the pathology. And why is this important? Because first of all, I think genomics was well characterized and well used over the past many years, but there is an increasing awareness that unless you go into the transcriptome, which is basically maybe a little more closer to the functional end of things, and the regulatory mechanisms which are brought about through epigenomic and mRNA studies, you cannot really have like a wholesome study completed. And so through that, these biomarkers have been interrogated, right? And there are also other things which are involved with head and neck cancers, like for instance HPV is a very common pathogen which is associated with the oral OSCC, or head and neck cancers. And interestingly, there are nice molecular tests available for a detailed genotyping for HPV. As you know, HPV is also associated with cervical cancers. So there are panels available commercially, which not just classify the genotype of HPV associated, but also concomitant infections with other STDs, right? Of course, they don't have any ramifications, node of cancers, but it has ramifications with cervical. Similarly, another omics that we can potentially talk about is the metagenomic approach, right? As we know, the oral cavity has the second largest and diverse microbiota after the gut and harbors over almost 700 species of bacteria, right? Now, aside of that, there are other classes of microorganisms which are associated with the oral cavity. That includes fungi, viruses, protozoa. As we know, during COVID times, I think, and something which made genomics a living room conversation was the fact that people were actually using the metagenomic studies from the oral samples for viral for actually figuring out in the initial days what kind of virus is involved in the infection. And that's how they chanced upon, through the metagenomic RNA metagenomic studies, they chanced upon the SARS-CoV-2 virus. And even after that, a specific test, sequencing test, was designed to study how the virus was mutating, right? Similarly, the oral microbiome diversity, uh, there is a baseline diversity, and then depending on whether or not you have some kind of a pathology, you will see a deregulation of that baseline diversity in those cases with diseases, right? And a very again, there is an ease of sample collection. So that's the reason why, along with gut microbiome, it has become the most studied microbiome till date, right? And these are some of the facts about the oral microbiome. I'll not go into the details of it, but it suffices to say that we have now very elegant methods for studying the oral metagenomics. And in the recent times, there is enough and more evidence to kind of really safe to be for us to be able to safely say that microbiome in the body really decides a lot of things, including whether or not you're going to come down with the disease, whether or not you have certain responses to certain diets, whether or not you have certain responses to certain drugs, etc., or your general well-being. It has been so gut microbiome, for instance, has been associated with a lot of you know disease response to drugs, the behavioral changes, the adaptation, etc. Similarly, oral microbiome has also been associated with a lot of pathologies. So the commensal population, or the symbiotic population in the oral cavity, in case of a pathology, uh, becomes deregulated, and that's when you actually that's nicely indicative of different disease source, right? So one of the ways that you can do this is through the 16S amplicon sequencing, which means, so basically what you do is you have 16S rRNA, which is amplified, and it has these 9 variable regions. So often sometimes people amplify all the variable regions. Sometimes people amplify the V3 and V4 regions. That's what's most commonly done. And the amplicons are studied through sequencing. So it's a simple amplicon sequencing method. And when you actually analyze it through certain tools which are available, like Kraken and all of these tools that are mentioned here, you're able to figure out the microbial diversity. Okay, now there are two um, methods to do it. You have the shortcut metagenomics method, in which you isolate the entire DNA, you fragment it, and then you sequence it. And there is a 16S method, where you essentially like amplify two two or a few more variable regions and study those variable regions which are indicative of different bacteria present in the population. And there are some recommended methods of sample collection and extraction of DNA, because a lot of times, when saliva may not be the best way of, like, a few people have also commented that saliva may not be the best, uh, you know, um sample, because a lot of times for some people, your DNA amount is often a question mark in the saliva. But you can include, say, things like buccal swabs, or you can mix buccal swabs with the saliva, and there are hacks and tricks to make sure that your DNA isolation from the saliva is efficient, and you're able to get a good amount of DNA for anything like a metagenomic sequencing, a 16S amplicon sequencing, or even a metatranscriptome sequencing, which basically helps you decode some of the functional pathways, and basically do like a functional correlation with the metagenome present, right? So all of this put together, essentially, it's safe to say that there is an ecosystem of multi-omics tool which is now driving research and clinical studies on various biological specimens. And what we believe is that the future will move towards these non invasive sample interrogation. When I say non invasive, even blood is actually non invasive, even though it involves a prick. But saliva is, I think, one of the most convenient samples to collect. So there is a huge industry of consumer genomics, which is also hinging on saliva collection and saliva genomic DNA extraction and saliva analysis, where so we've spoken about, the analysis of DNA in pathology, in different pathologies, or we've discussed about the oral microbiome sequencing or metagenome sequencing. But there's an entire industry which is hinging on the DNA sequencing of the host, or the DNA analysis through microarrays for the host, wherein you can actually tie it to wellness. So whether or not my genomic DNA predisposes me to a bunch of diseases, whether or not my genomic DNA has those variations which will make me respond to certain drugs better, or whether or not my genomic DNA has those variations. These are called pharmacogenomic variations, which will basically decide the way I respond to different drugs. There are whether or not the variations in my DNA will actually decide whether or not I metabolize certain nutrients. Like, I decide my diet plan based on the genomic DNA I have, and the variations in it. So all of this is a part of a wellness industry, which now analyzes DNA routinely and hands out wellness reports. So you might have heard about 23andMe, which also did like this ancestry thing where and the preferred sample collection is saliva, right? We have a bunch of companies in India also which are handing out reports to people. And like I said, the preferred sample collection is saliva. So, yeah, I mean, basically, when you talk about technologies, we have a bunch of technologies which help you gain insights from the biomolecules which are collected, or the biospecimens which are collected from different individuals. And these help to not just analyze the DNA, but also the transcriptome, the proteome. You have single cell genomics, you have technologies which give you disease or oncology predictions, etc. So there's a nice ecosystem of technologies existing which will help you reach that end that you want to reach. And if you want insights into some of these technologies, I mean, we're there. You can contact us. And I mean, I think we're all existing in a space which is very exciting, and we can really work together to make sure that meaningful outcomes are generated through exercises that we undertake. With that, I think I'll, like probably wind up my talks. Thank you so much for listening to this little presentation that I put together. And I'm happy to take any questions, which might be there. Thank you.
Thank you, Dipjani. It was a fantastic session. And I think as of now, we don't see any questions in the comments. Like, I would like to ask one question as you were saying, since you are in a space from the industry in genomics for a very long time. So, one question that runs in my mind, like when you were talking about, the genomics NGS market as such, and what we see that a lot of interest is inside the clinical space now. So, one question as an educationist I would like to ask is that uh, uh, is like, how do we bridge the gap between the understanding of this technology from technical perspective and clinical perspective, meaning to say that is it necessary that a physician or a clinician really need to go through the understanding of these technologies, because ultimately it's healthcare professionals who are going to recommend patients through this. So that's a question which runs in my mind. If you can throw some light on that.
So I think thanks, Gargi, and I think it's a very important question because I think the clinical utility or the utility, see today, genomics is no more a technology which is like desired for in the laboratory. We're looking at genomics as a means of diagnosis. Many years back, that was the status with MRI or PET scans, etc., which have now become commonplace, really, right? So, if we have to make genomics a little more mainstream, because we feel that it has this value add that it can do for helping us reach the reason, I mean, basically helping us to unravel the reason of any pathology. If we have to reach that stage, we'll definitely have to have a clinician awareness. The clinician awareness should be on the scope of the technology in terms of what they get to benefit from, right, and not necessarily the granular details on the workflow, right? So I won't say that the clinician needs to understand what the workflow should be, or how do you put up a reaction, or how do you put up an experiment. I think the work has to be divided. There are people to take care of that. I think they should be able to see the advantages that the technology brings to them in terms of the added benefit that it gives to the entire treatment exercise. So if suppose a patient comes to me and I'm a clinician, I should be able to figure out the utility of a next generation based test to the patient. In certain cases, it may not add value. In certain cases, it just may not really bring a real advantage. But in those cases where it does bring an advantage, I think its scope should be at least analyzed. People should at least figure out what it can do, because I think ultimately it has to be an informed. Suppose if I decide treatment regime for my patient, at least that treatment regime should be well informed. I should have all the facts, in front of me, and especially, certain kind of genetic disorders or certain kind of cancers where, the disease itself is very complex. And, a lot of times the plain, archaic based testing may not be able to add a real value. I should be able to at least have the facts clear in front of me through the genomic forays. So I think it's that basic education needs to be there. So I think we should definitely think of integrating maybe some kind of a genomics, credits added to medical professionals course curriculum, because unless they understand that, the good thing is there are a lot of companies which are now doing these continuing medical education, the CMEs, as we'd like to call them. And that's an endeavor which has been taken over by multiple companies. However, like I think we need more foot soldiers to do that. And also, I think there should be integration of all of this into the course curriculum of the clinicians.
Thank you, Devjani, for
Thank
that.
you, Devjani, for that. Yeah.
And we'll wind up for this session. And it's a pleasure. It was a pleasure having you here. Thanks a lot.
Thank you so much. Thank you.